Hello students, today we are going to learn for Introduction to Biochemistry. So this is first lecture of biochemistry and learning objectives for this lecture are students will understand what is biochemistry, they will know the components of a cell and its types, understand their functions. So let's go through all the topics one by one. What is biochemistry? Biochemistry is the application of a chemistry to the study of biological processes at cellular and molecular level. It emerges as a distinct discipline around the beginning of the 20th century when the scientists combined chemistry, physiology, biology to investigate chemistry of the living systems by studying structure and behavior of the complex molecules found in biological material and the ways these molecules interact to form cells, tissues, and whole organism. So, next one is the principles of biochemistry. Cells are highly organized and constant source of energy is required to maintain ordered state. Living processes contains thousands of chemical reactions. Precise regulation and integration of these reactions are required to maintain their life. Very important reactions like glycolysis are found in almost all the organisms. All organisms use same types of molecules, carbohydrates, proteins, lipids, and nucleic acids. Instructions for the growth, reproduction, and development of each organism are encoded in their DNA. So what are the applications of biochemistry in pharmacy? Biochemistry has significant importance in pharmacy. First application is drug constitution. Biochemistry helps in constitution of new drugs. It helps to improve the efficacy of a drug and minimize its side effects. Second one is half-life. Half-life of a drug is the time to take for the drug active substance in the body to reduce by half. It means drug half-life will depends on how much time it will take to degrade by its half. Biochemistry helps to determine half-life of the drug. Next application is changes of the biodegradation of the drug. Biochemistry helps to determine stability of a drug by calculating changes of its degradation with changes in the atmospheric conditions like temperature, humidity, etc. It also helps to determine the expiry date for the drugs. Next one is drug storage. Obviously, biochemistry can help to determine the storage conditions of any drugs. Drug metabolism. Biochemistry gives the idea about how the drug molecules are metabolized by many biochemical reactions in the presence of enzymes. Next one, detect nutritional deficiencies. Deficiencies of biomolecules such as proteins, lipids, vitamins, minerals, and water may cause diseases. Biochemical research has a role in preventive medicine by helping identify such diseases. Disease understanding. Health depends on the proper balance of biochemical reactions occurring within the body. Abnormalities in biochemical reactions result in diseases. Biochemistry helps to understand diseases like protein, energy malnutrition, inborn errors in the metabolism of carbohydrates, proteins, lipids, etc. So now we go towards the next topic, that is a cell and its biochemical organization. You already know that cell is the basic unit of your body and cells eventually forms a living body. So what is a cell? So cell is defined as a basic living structural functional unit of an organism which is capable of carrying out the processes of a life independently. Cells are the basic building blocks of life. They grow, reproduce, use energy, adapt and respond to their environment. The concept of a cell originated from the contributions of Sheldon and Schwann in 1838. However, it was only after 1940 that the complexities of the cell structure were exposed. So what are the importance of the cell? So it is a basic unit of structure and functions in the human. It contains diverse subcellular organelles specialized for various functions. Diseases can arise from organelle dysfunction. Cell cycle governs growth and division. Molecular composition of a cell. So there are three major components of a cell, that is water, organic compounds and inorganic compounds. So around 70 to 75% of a cell weight is composed of your water. 25 to 30% compounds are of organic compounds, including proteins, nucleic acids, carbohydrates, lipids. Inorganic compounds such as electrolytes, which are vital for your cellular homeostasis, are present and as a molecular composition of the cell. So there are different types of cells, that is prokaryotic cells and eukaryotic cells. Prokaryotic cells include bacteria, and they lack basically nucleus membrane bound structures called as organelles. Eukaryotes include most other cells and have nucleus and membrane bound organelles. For example, plant, fungi, and animal cells. So these are the key differences of your prokaryotes and eukaryotic cells. So prokaryotic cells are always unicellular, while eukaryotes are unicellular and multicellular. Cell size ranges between 0.2 micrometer to 2 micrometer, while in eukaryote, the cell size ranges from 10 micrometer to 100 micrometer in diameter. As already mentioned, in prokaryotes, nucleus is absent, while in eukaryotes, it is present. Ribosomes are also present in both eukaryotes and prokaryotes. DNA arrangement in prokaryotes is circular, while in eukaryotes is linear. Mitochondria are absent in prokaryotes, while in eukaryotes, mitochondria are present. Reversely, endoplasmic reticulum is also absent in prokaryotes and present in eukaryotes. And examples for the prokaryotes are bacteria, and examples for eukaryotes are animal cells, plants or animal cells. So structure of prokaryotic cells, as already we have discussed in difference of prokaryotic cell and eukaryotic cell, we already know that it does not have a nucleus membrane. Genetic material is present in the cytoplasm in a region called nucleoid. Prokaryotic cells lack membrane-bound organelles. These cells are generally small and simple in structure. Prokaryotes may be spherical, rod-shaped, or spiral in form. Prokaryotic organisms are divided into two domains, that is bacteria and archaea. So this is the structure of your prokaryotic cell. As you can see, it has a capsule, cell wall, plasma membrane, cytoplasm, and nucleoid, which is mentioned, and circular DNA, which was already mentioned during the difference. So next move towards, so the components of your prokaryotic cells are capsule, cell wall, cytoplasm, cell membrane, that is plasma membrane, pilus singular, that is a hair-like structure on the surface of the cells that attach to the bacterial cell, flagella, ribosomes, plasmiodes, and nucleoid region. So nucleoid region, that is single bacterial DNA molecule which is present. Ribosomes, they are cellular structures which are responsible for protein production. Next one is eukaryotic cell. As you can see, all the components of the eukaryotic cell, that is, it has an outer cell membrane, lysosome, microtubule, cytoplasm, ribosomes, plasma membrane, nucleus, Golgi complex, vacuoles, mitochondria. So we will see all the components one by one. Cell membrane, that is a plasma membrane. It was proposed by Singer and Nicholson in 1972. The cell is separated from its surrounding by the thinner membrane called as a cellular membrane or a plasma membrane. It is composed of 40% lipids and 60% proteins. The cell membrane consists of a double layer of phospholipid molecules. Proteins present in the cell membrane provide structural support, form channels, form for movement of substances. There are different cell membranes proteins, intrinsic proteins, which are embedded within the cell membrane. So it means they will be already inside your cell membrane. Next one is extrinsic proteins. They will be attached to the outer surface of your cell membrane. So below are the various functions of your cell membrane. That is, it encloses and holds the cell. It acts as a selective barrier. It allows specific substances such as nutrients to pass through. It helps in secretion of waste products. It prevents entry of harmful toxic substances. It protects internal components of the cell. Next part is, so we already seen the cell membrane. Now next part is a nucleus. Inside cell there is a central dense body called as a nucleus. which is separated from your cytoplasm by your double-layered membrane that is called as a nuclear envelope. The inner and outer membranes of the nuclear envelope join at certain places to form nuclear pores. These pores allow exchange of materials between the nucleus and cytoplasm. The living substances present inside nuclear membrane is called nucleoplasm. The nucleoplasm contains nucleolus, which is rich in RNA, DNA, or other genetic materials. It solely contains all the genetic materials. The major portion of the nucleoplasm consists of a thread-like structure that is called chromatin, which is consists of your deoxyribonucleic acid. So what are the different functions of the nucleus? So cell nucleus is the core of your cell. It controls and regulates all the functional activities of your cell. It contains chromatin, which is most important genetic material. Chromatin condenses from the chromosomes, which is equal distributed daughter cells. It is a site where DNA-directed RNA synthesis plays essential role for your protein synthesis. Mitochondria. Mitochondria are the rod-like large components of the cytoplasm of the cell. Each cell contains 15 to 2500 mitochondria. They consist of a proteins, lipids, small amount of nucleic acid. It is composed of a double membrane system. Energy producing chemical reactions takes place in crystal. The inner chamber is preferred as a matrix or mitosol. The functions of mitochondria, it plays important role for the respiration and energy production. So it is also called as a powerhouse of the cell. It is responsible for transportation of a chemical energy into biological energy to form the ATP. They are responsible for transmission of the hereditary characters. Below is the structure of your mitochondria. Next one is endoplasmic reticulum. Within the cytoplasm of the cell, layers of the cytoplasmic membrane are folded inwardly to form a network of the vesicles and tubules near the cell surface. It is called as the endoplasmic reticulum. There are two different types of the endoplasmic reticulum. That is granular endoplasmic reticulum. It has ribosomes attached to its surface. These ribosomes are responsible for your protein synthesis. Next one is a granular endoplasmic reticulum. It is involved in the synthesis of your fatty acid. It means there is a difference between granular endoplasmic reticulum and a granular endoplasmic reticulum. There are different functions of your endoplasmic reticulum. The rough endoplasmic reticulum, that is granular endoplasmic reticulum, is involved in the protein synthesis, while the smooth endoplasmic reticulum is concerned with the lipid and glycogen synthesis. It also provides a mechanical support by forming skeletal network. The enclosed spaces of the endoplasmic reticulum helps in exchange of materials between cell and extracellular fluid. Next part of component of your cell is Golgi body. Golgi apparatus is also known as a Golgi complex. They are the membrane-bound organelle composed of a series of flattened sacs, and it involved modification, sorting, packaging, transport of proteins and lipids. There are different functions of your Golgi body or Golgi apparatus. It is also act as a site for enzyme secretion, participants in the formation of lysosomes. It plays important role in the membrane synthesis, especially for the formation of the intracellular organelles. Lysosomes are membrane-bound organelles. They are responsible for the breakdown of the proteins, lipids, and damaged cells components. Lysosomes also helps in the digestion of the materials indigested by the cells, hence also known as suicidal bags, and below the structural representation of your lysosomes. Next one is cytoplasm, very important component of your cell. The cellular matrix is collectively referred as cytoplasm. Cytosol is the aqueous gel-like portion of the cytoplasm that contains enzymes, metabolite salts. Functions of your cytoplasm, it helps for maintaining osmotic pressure inside the cell. So now next move towards your question answer sessions. So I hope you have understood all the components of your cell, understand the definition of a biochemistry. Let's see how you are able to answer the question. So, which cell organelle is known as a powerhouse of the cell? Can you please tell me? Yes, correct. It is mitochondria. It is called as a cell powerhouse of the cell. Next one is match the pair. Here are the different organelle and different functions of your cell components. Ribosomes, Golgi apparatus, mitochondria, and their functions are ATP production, protein synthesis, and packaging proteins. So ribosomes. So which one you can answer first? Yes, correct. For mitochondria, you can have a ATP production. For ribosomes, you can have a protein synthesis. And for Golgi apparatus, it is a correct one. It is for the packaging of proteins. I hope you understood the lecture 1 very clearly. We will come again with a lecture 2 with a new topic. Thank you all.